All right, guys, so welcome back. We're still in Unit 2, Humans versus Bacteria. And so far, we've gone through two of our four 5E segments for the unit. The first one, if you remember, was the Black Death, which was all about the bubonic plague, which was also another very, very infectious, contagious disease that was spread by a bacteria, a pathogenic bacteria. Remember, a pathogen is anything that causes disease, right? So it could be a bacteria, it could be a virus, it could be some type of like fungus, it could be some type of worm or parasite or something, but anything that causes harm to the body, disrupts homeostasis, causes disease or disorders, right? And we spoke about why it was so deadly and what humans did historically to try and combat the bubonic plague. And then we moved into antibiotic resistance, which was really talking about how humans have developed antibiotics to help fight off diseases such as cholera, right? Because we really want to prevent another cholera outbreak from occurring. That's the overarching focal point of the entire unit. How can we prevent another outbreak of cholera, right? Because if you remember from the unit opening, we've had seven, and we're currently in one called El Tor in Egypt, right? Which has lasted for quite a few decades now. And that was really just talking about how bacteria have now resisted almost every antibiotic that we've been giving them to kill them. So the overall effectiveness of antibiotics over the years have gone down. And it's likely because we've been overusing them and misusing them. So bacteria, they're kind of onto our game now. Like, we know what you're going to use to try and kill us, but we have our own mechanisms to resist that. And if they're able to resist that, then guess what they're able to do? Survive. And if they're able to survive, then they're able to reproduce, right? And likely pass on those genes of resistance to future offspring. Now we're going to move into the microbiome, which is a very important 5E learning segment because it really talks about the importance of bacteria, especially as it pertains to human health, right? Believe it or not, you would not be here on Earth if it wasn't for bacteria. All right. And we're going to explore in this 5E why that is. So, our new 5E learning segment is called the microbiome, and this is our engage lesson. And our aim says, why would we be able to treat a bacterial infection by introducing more bacteria? That's an interesting concept, but let's deep dive in. How can we treat a bacterial infection? Let's say it's maybe cholera by introducing more bacteria. That sounds counterintuitive. Why would I introduce more bacteria if I'm trying to treat a bacterial infection? That leads us into our do now, which says, are all bacteria harmful? Why or why not? And the short answer is no, no. All bacteria are not harmful, right? In fact, most bacteria on Earth are not harmful to humans and may actually benefit us, right? Now, I'm sure you're wondering, how can bacteria benefit us? Well, they can in a plethora of ways, actually. So some help us digest certain foods. Some help to fight slash kill off harmful bacteria. And some even produce certain enzymes to help us with certain metabolic functions. right? So with certain life processes, right? So bacteria are very, very, very helpful. And in fact, we have a very close symbiotic relationship. And we'll talk about what symbiosis means. So part A, fecal transplant. I know that sounds absolutely disgusting, and I honestly wouldn't blame you if you thought that. But let's actually break down what this means, a fecal transplant. Now, if we look at the word fecal, we should see the word feces, and feces is just your undigested waste, right? So when you need to number two in the bathroom, you're releasing your feces. And then transplant, I'm sure you're wondering, where is this going? Undigested waste transplant, right? A transplant just means taking from one area to another, right? We're transplanting it like an organ transplant. So a fecal transplant. So let's take a look at our initial thoughts. It says, why and how do some doctors treat bacterial infections by introducing more bacteria? And I want you to pause the video here. And these are going to be your initial thoughts. I'm not going to write anything here because then those would be my initial thoughts. But I want you to really read the question. I'll read it again with us. Why and how do some doctors treat bacterial infections by introducing more bacteria? So it's just getting your initial thoughts on what the AIM question was above, which was why would we be able to treat bacterial infections by introducing more bacteria? So pause the video here and jot down some initial thoughts about why doctors would treat a bacterial infection. Think of like cholera. Why would they do that? By introducing now more bacteria. Okay. And then once you've taken 3 minutes to write down your initial thoughts, I want you to unpause the video and we'll continue with our reading and annotations. Okay. All right. Now that you have your initial thoughts written down, let's read. So, a new treatment. Patients who are hospitalized often or live in nursing homes sometimes get a nasty and recurring infection from a bacteria called Clostridium difficile. And we're going to shorten that to C. diff, right? So now they're telling us about another type of really nasty pathogenic bacteria. We already spoke about cholera in the unit opening and how nasty that was, and how we're still in a cholera pandemic worldwide and that it keeps coming back and causing all of these nasty, severe dehydration with diarrhea and vomiting, and it affects the small intestine. Then we spoke about the bubonic plague, and it has like those large buboes, and people just literally look like the Walking Dead, lots of muscle fatigue, and it spread from person to person, and it was really just taking out most of Europe. Now we're talking about a type of bacteria called C. diff or Clostridium difficile, which is sure to affect a lot of hospitalized people and a lot of like the elderly who typically might live in nursing homes. So let's read a little bit more. People with C. diff infections can suffer severe colonic inflammation, right? So your colon is really just another word for your large intestine, and it's being inflamed. It's not going to be great, which could lead to symptoms ranging from recurrent diarrhea, which could last from weeks to months, to kidney failure or even death, right? So if we have recurrent diarrhea, our body is literally getting rid of a lot of water, right? So this leads to dehydration of the body. And as we learn with the marathon runner, the kidneys are what's responsible for osmoregulation, right? To balance out the amount of water, to produce urine for us. But if your kidneys are failing, can you get rid of those waste products such as water, salt, and urea? No. And if those waste products build up in your blood, you can get a nasty, nasty, deadly infection. C. diff can really win this battle, and this could lead to death, as the text suggests. Let's continue. C. diff or C. difficile is responsible for about 29,000 deaths annually, right? So every year, on average, 29,000 people die from this infection. And administering antibiotics often does not cure these patients, right? So here we have another example of how this bacteria is resisting the effects of the antibiotic that's used to kill them, right? It's saying even if we give antibiotics to these patients, it's not helping them much at all, right? And it could be because C. difficile is just saying, haha, I've already evolved to resist this antibiotic, try again. Now, due to this, researchers are constantly on the lookout for better ways to treat the infection, right? So they can't rely on antibiotics because they're not working. So we're constantly looking for ways to try and treat the infection and lower the risk of death. Research coming from the University of Texas Health Science Center at Houston may lead to just that in the form of a pill. So now we're really curious. But what is inside that pill? What is inside that pill? Fecal matter from a healthy donor. Ooh. This process is called fecal matter transplant, excuse me, and it involves extracting healthy bacteria from another individual's fecal matter and then processing and transferring that bacteria to the infected patient directly, right? So someone that's very healthy, they might take the bacteria that dwells in their fecal matter. Oh, yes, you have lots of bacteria that's in your fecal matter. Because remember, your feces is waste to you, but other organisms love it. Think about what flies and maggots they plant or they lay their eggs inside of feces. Why? Because there's a lot of nutrients that our body didn't take in, that our body disposed of in feces that's very beneficial to other organisms. In fact, what do you think that we grow a lot of our fruits and vegetables in? A lot of our plants that we eat is grown in manure, right? And that's because it's very rich in nutrients. So bacteria, they're going to take advantage of that too inside of our bodies. A lot of it is going to pass out with the feces. So if we can extract the bacteria from a healthy person's feces, right? These beneficial bacteria, we can then process them so that way they're behaving properly, and then transfer them to the infected patient directly, right? This patient that is suffering from the C. difficile infection, we can introduce healthy bacteria from the person's feces. All right. Frozen or freeze-dried bacteria from fecal matter can be administered in a pill form at a patient's or doctor's convenience, whereas fresh transfers require a donor and recipient to spend hours in the hospital to obtain the fecal sample, refine it, and then deliver it to the infected person, right? So what's the benefit here? We can literally extract these healthy bacteria from the healthy person's feces or from their fecal matter. And we can freeze dry them, right? To where we freeze them where they don't die, they just remain dormant, right? They're not active. And then we can process them in the form of a pill, right? Just like you would take maybe a painkiller or just like you would take maybe an antibiotic. You can take this healthy bacteria in the form of a pill, right? Um, and you can take it anytime. That's the benefit of that, right? They also can do a fresh transfer where they literally take the donor and the recipient, But the donor and the recipient both have to be there. They have to spend hours at the hospital to obtain the fecal sample, wait for it to be refined, and then deliver it to the patient directly. It's going to take a lot more resources than just literally popping a pill, right? So doctors prefer giving it in pill form than to do the whole okey doke of a fresh transfer, all right? But that makes it very convenient that we can just take it in the form of a pill. Let's conclude. So, one of the participants in the Texas Health Science Center study is already sold on the new method of treatment. All right. He excitedly told a reporter that his cure is a miracle, right? So he did this fecal transplant and he's saying, oh my gosh, this is the best thing ever. My cure is a miracle. Let's see why he said that. He added that prior to the new treatment, so before he did this, he had been considering retiring from his teaching job, go figure, because the infection had made him so ill, he wasn't always able to do his best work. So he was literally suffering from this C. difficile, right? Causing him to have possibly kidney failure and constant diarrhea, right? And he said, I love teaching, and I can't tell you what it was like to be healthy again, to be able to do my job and not retire before I was ready, right? So this entire anecdote, this entire story that's here is evidence that supports fecal transplants, right? We have one participant that's literally raving about it. It worked very, very well for him. Now, is this strong evidence or more weak evidence? We would say that this is more weak evidence that supports. Why? Because the sample size is one. This is one person. It's not like we're saying 3 million people from the study are all raving and ranting about it. That's much stronger evidence. But this is just one person, right? And we know with any experiment, in order for it to be nice and strong and valid, we need to have a much larger sample size than just one. So it is evidence that does support it, but it's all anecdotal. This is the story of one person. So even though it may have worked for him, it doesn't mean that it'll work for everyone. Now, our final thoughts. Why and how do some doctors treat bacterial infections by introducing more bacteria, right? So we already had our initial thoughts, but let's write down what some of our final thoughts are now that we're equipped with more information. Well, this involves taking bacteria from fecal matter of a healthy donor, freeze dry them, right? So that way they're not dead, but they're just inactive, and make an oral pill. Now, the whole purpose is that these healthy bacteria can help fight infections such as C. difficile by restricting and or outcompeting by restricting or outcompeting them, the C. diff, for essential resources needed to survive and reproduce. This may potentially decrease dependency on antibiotics. So they already told us that antibiotics are really not working. So we need another plan of attack to help these people. And so far, the plan of attack is to take these healthy bacteria from fecal matter from a healthy donor, freeze dry them, and give this oral pill that have these healthy bacteria to the person that's suffering from this infection. What we hope, right, is that those bacteria, once they've thawed out, they'll be able to now restrict or outcompete this infectious bacteria, C. difficile, for resources that they need, such as nutrients, space, all of those different things. Nutrients and space is going to be the two most important essential nutrients or essential resources that are needed. And if they are able to outcompete, then the healthy bacteria are going to survive and reproduce, right? And the C. diff population is going to decrease, which is exactly what we want, right? Because they're no longer getting as much nutrients and as much space as they need in order to survive and reproduce. So they're going to decrease. Those populations are going to decrease, right? So this is how scientists are now using bacteria to help us fight off bacterial infections. We're starving the infection of what it needs in order to reproduce and to survive in that environment. If we just leave it alone, then the C. diff is just going to like take over and sap all the resources out of it. You can think about it as like weeds in a garden, right? The reason why when people have a garden, they try to get rid of weeds as quickly and as efficiently as possible is because weeds are very dangerous. They absorb nutrients from the garden, which means that other plants have to starve. They're not getting the nutrients that they need because the weeds are taking all of them. They're a lot more efficient at taking those nutrients, right? So they're better adapted to that garden area. And we don't want that because those weeds are eventually going to starve the actual crops that we want to grow, the actual plants that we want to grow of what they need. And so if they're being starved of their nutrients, they're not going to be able to survive and reproduce. In fact, no organism that's starved of its nutrients are going to be able to survive and reproduce. So we have to remove those weeds. You can think of the C. difficile infection as the weeds of the garden inside of the body, right? And we need to introduce healthy bacteria that are able to outcompete them for those resources so that way they don't win the fight. Okay? All right. So we are making our way down to part B. Now, if we take a look at part B, it says microbiomes and different lifestyles. All right. So we saw this word microbiome already, but we have yet to actually define it. And if we look at the word microbiome, it's broken up into really just kind of two words, micro and biome. Micro means very small, right? Think of a microscope. The whole purpose of a microscope is to view small things more largely, right? At a scopic level. A biome is just literally an area in which organisms live, right? So an area in which organisms live, right? So, and if you look at the word bio, which is in biome, bio means life, right? So organisms live in biomes, right? But we're talking about a very small organism. So we're talking about where bacteria live and where they thrive whenever we're talking about microbiomes, okay? Now, microbiomes and different lifestyles. Let's continue. List different things people do that might impact the bacteria that live in and on them. So by now, we know that bacteria thrive on us and inside of us, right? We provide them with a place to stay And a lot of those healthy bacteria help us to fight off potential infections that are trying to get through our skin and get through our mucous membranes to come inside of our body and wreak havoc. They help fight off those things. They help to provide us with vitamin K inside of our intestines, otherwise, that we would have a difficult time actually getting. They help us to digest food, all of those things. But the number one thing that people do, right, or that they engage in that might impact the bacteria that live in and on them is potentially diet. If you ever go to the doctor, right, the doctor, no matter what type of physician it is, no matter what specialist the doctor is, no matter what practitioner the nurse is, or physician assistant, they always ask you, how's your diet? Do you exercise? What are you eating? And the reason why is because what we put inside of our body matters, right? How we take care of our body matters, right? The type of nutrients that we have inside of our body is going to determine how healthy our body actually is. And when we talk about how healthy it is, it just really means how well we're maintaining homeostasis and how well we're feeding those healthy bacteria so they can continuously help us to maintain homeostasis. That's what we care about. Another thing are medications, right? Such as antibiotics, right? If we use antibiotics a lot, we could be helping the bad guys win. We don't mean to, but that's what we could be doing without even really realizing it. And that's what we're actually seeing historically. That's why we see things like cholera coming back again, because these overuse of these medications to fight off these bacteria is wreaking havoc, right? But remember, antibiotics don't just kill the bad guy, it also can target the good guys. So when we use antibiotics, we actually help kill healthy bacteria, which is not great. Okay. We can also count geographic locations, right? Where people move to, where they live, right? Where you live, where you move to is going to dictate what type of foods you eat, what type of pathogens you're exposed to, what type of healthy bacteria you're exposed to, right? Your culture has a huge impact on the type of bacteria that live on and inside of you. All right. And then the last one, I'm going to leave up to you. I want you to come up with something and you're going to fill that in yourself. Okay. Now let's take a look at number 2. Now it says, what do you think our microbiome, which is all of the bacteria and other microorganisms that we host, was like before things like antibacterial soap and antibiotics were created? Right? So antibacterial soap is just special soap that contains antimicrobial agents that help to really kill those bacteria that are like on your hands and such. Okay. And then, of course, antibiotics we know are used to kill bacteria, both good and harmful bacteria that it kills. Right. So it's just asking us, what do we think that our microbiomes were like? What do we think our bodies were like with the type of bacteria that live on them was like before the use of antibiotics or the creation of antibiotics and antibacterial soap? Right. So let's speculate, right? In my mind, there may have been, or rather, so we may have had a more diverse microbiome, right? And the word diverse just means variety, right? So we may have had a variety, a much higher variety of bacteria living on us and inside of us, right? antibacterial, soaps, and antibiotics kill harmful bacteria, but they also kill beneficial ones as well. So as a result, this directly reduces microbiome diversity as seen today, right? And we can't forget to highlight evidences as being causal or correlational. So if we look here, it directly reduces microbiome diversity, right? The use of antibacterial soaps and antibiotics directly reduces. This is definitely causal. It tells us that it directly reduces microbiome diversity, right? So if our claim is that the use of antibiotics, just like we saw with the previous 5E on antibiotic resistance, the use of antibiotics directly impacts our microbiome, then yes, right? Because it's literally killing off not only just harmful ones, but also beneficial ones, which means that our diversity is going to be a lot less, okay? You can almost kind of think of it as like bacterial genocide, if you will. Now, number three says predict how bacteria that live in and on us may be different between the following communities. All right. If we take a look here, it says hunter-gatherers or people that live in isolated indigenous communities, also known as Hadsas, people that live on farms in rural areas, and people that live in the city. All right. So these are three different areas that we're essentially looking at here. Hunter-gatherers, rural areas, and people that live in the city. So let's start with hunter-gatherers, also known as the Hadsas. Those are going to be interchangeable. All right. And then we'll move on to rural areas and then people that live in the city. So it tells us that they live in isolated indigenous communities, right? So in my mind, when I think this, I think that they may have bacteria that are exclusive to their microbiomes due to isolation. So because they're isolated from the vast majority of other societies, they're not coming in contact with many people. Therefore, they're not exchanging bacteria from like their hands or from like their skin to skin or like sharing food and drinks and things like that. So they may have bacteria that are, they're only living amongst themselves. So they may have bacteria that are very exclusive to their microbiomes. In other words, I wouldn't go somewhere else and find these certain bacteria of people that were not hunter-gatherers because they're living in isolation. Okay. Now, another thing that it makes me think is they might have perhaps the lowest microbiome diversity slash variety due to isolation from other groups of people. I'm not saying that it's right or wrong, but in my mind, that's what it makes me think, right? Because the question is asking us to predict how bacteria that live in and on us may be different between these three communities. So, just due to the fact that they're isolated indigenous communities, I would think that they maybe have the lowest microbiome diversity because they're so isolated from other people or from other sectors of the world, right? So they're not getting as much bacterial exposure, if that makes sense, to other varieties of bacteria because they're so isolated. And then it also makes me think that perhaps they have the highest microbiome diversity slash variety. They mean the same thing. Since they are much less likely to utilize slash benefit from medical advancements in modernized societies, such as antibacterial soaps and antibiotics. So I know I basically just said the opposite of what I said in the second bullet, but I have two ways of thinking about this, right? So in the second bullet, I said that it may have the lowest microbiome diversity because they're so isolated from other, societies of people. But that might also be the benefit is the fact that they are isolated from other and they're not utilizing antibiotics and other medical advancements nearly as much as more developed societies, quote unquote, developed societies might be, or other societies that live in a city where having access to adequate healthcare is the need for it is much higher. In like, let's say a place like New York City, where the population is so high and the rate of transmission of diseases is also so high, right? We need to take advantage of antibiotics, vaccines, all of those things. But if I'm living in isolation, the need to take advantage of those things is a lot less because I'm not being exposed to so much more as someone living in, like, let's say, a city. So it makes me think both ways. Now, these are just what my thoughts are, but I want you to write down some of whatever your thoughts are as well. And again, it says that it's a prediction. So you absolutely cannot be right or wrong, just be able to rationalize or reason, right? Claim evidence, reason why you think the way that you do for each specific peoples. Now, if we move on to people that live on farms in rural areas, it makes me think that there might or that they might have bacteria in their microbiome found on livestock and agriculture in the area. So in my mind, they're going to have bacteria that might almost be exclusively found on certain animals, certain livestock, such as maybe cows, you know, bulls or other cattle that they're harvesting, and also bacteria that almost exclusively like live on plant life, right? Because they're interacting with plants and animals regularly. So naturally, those bacteria are going to migrate from the livestock to the person. Then it also makes me think that the microbiome diversity might be lower than the hunter-gatherers. I'm just going to put HG, since they may need antibiotics for their agriculture to increase supply of products. If I'm a farmer, just like we saw at the beginning of the antibiotic resistance 5E, the previous 5E, we watched that video about how farmers use literally 70% of all the antibiotics that are sold in the US, right? And why was that such a great need for them? Well, it helped to keep their animals alive and lower the risk of infections, which means that they can harvest more cattle. More cattle means more meat, more product. More product means more profit, more money, right? So, in my mind, because they have a higher use of antibiotics in general, I would think that the use of those antibiotics killed off a lot of their microbiome. Maybe not all of it, but a lot of their microbiome is killed off. Versus the hunter-gatherers, they probably are not engaging with these antibiotics at all due to their isolation. But when you live on a farm, you might be isolated, but you own a lot of land, right? And there are going to be like interactions between people that live on the farm and don't live on the farm. So they're coming into contact with, on average, more people, right? So their risk of transmission of certain diseases, certain bacteria that might be harmful is going to be higher than hunter-gatherers, prompting the need for the use of antibiotics. And then finally, people that live in a city. So if we take a city like, let's say, maybe New York City, that's the city that never sleeps, right? I would think that it's the most modernized, and therefore may use slash take advantage of advancements in technology and medicine the most. So of the three areas, I would think that in a city, they're taking advantage of the use of technology and medicines the most. In rural areas, they're taking some advantage of it. The hunter-gatherers are taking very little advantage of it, right? So if that's true, then that would also lead me to believe that of the three, they have the lowest microbiome diversity of the three groups, and that most people, wait, sorry, not most people, sorry, and that they are the most prone to illness and diseases. There would be no need for us to be using so many antibiotics in the city unless we were more prone to illness and diseases. Otherwise, why would we be using it if we're not getting very sick? And if you live in a place like New York City where the sanitation is not the best at all, right? There's lots of like vermin, such as rats, mice, cockroaches, right? We use the subway, we interact with what I think the population now is like 8.5 million people. We are exchanging bacteria and exchanging diseases like never before, right? So therefore, we're much more prone to picking up something that's harmful than, let's say, the hunter-gatherers that are just living amongst themselves and not really interacting with the rest of society. Okay? So that is our engage lesson on fecal transplant and the microbiome, and really just starting to see how different areas, right, take advantage of these technologies, but also what the consequences are of taking advantage of these technologies as well. Okay. So after this, we are going to have an explore, which is going to compare a microbiome, comparing microbiomes investigations of three different groups, which we already kind of started doing here. But these were just predictions. We just speculated. Now we're going to look at some charts, analyze some graphs and some data to see if what we predicted was supported or refuted, and maybe why that was the case. Okay? You guys are absolutely amazing, and I will see you next time. Bye.